thank you to Dysphonia International. Thank you to Kim, Charlie for organizing this. Thank you for the opportunity to meet with patients. As Elijah said, this is really what allows us as clinicians, as researchers, as educators to really understand what the needs are and to really go after that. And so that's the goal here, and that's the purpose of us all coming together, learning together, and getting better together. So I appreciate that opportunity. I am a neurologist, so my bias is thinking about brain function and how that might be contributing to voice disorders. So I'm going to talk a little bit about that. I want to emphasize the role of patients, right? Everyone here in scientific advancement. Charlie alluded to this, and so we're going to talk a little bit about that. We're going to describe briefly normal voice control. I know we've heard it. I'm going to emphasize the role of the brain function in controlling the voice and how dysfunction of the brain may contribute to voice dysfunction. I'm going to highlight our ongoing research with respect to that supports network level brain dysfunction in spasmodic dysphonia. And then based on this, we're going to emphasize the future potential for therapeutic gain, better treatments. This slide's critical. This is what Charlie mentioned in the introduction today. Us being here together matters, right? So the mission is, as indicated by Charlie, is dedication to improving the lives of people affected with spasmodic dysphonia and related voice conditions through research, what I'm going to present today, education, what we're doing today, awareness and support. And this slide here really represents this, right? So this is publications in research is really the currency, right? You know, success in science allows us to disseminate knowledge to others. And so this here is the number of publications over time between 1900 and 2021 in this plot. These are, again, in my biased approach, dystonia publications, right? Not published until patient advocacy groups were developed, the DMRF and NSDA, now Dysphonia International. And this is not a coincidence that you see an exponential growth here. This is the seed funding that Charlie discussed that allows researchers like myself in medical school, in fellowship, in training periods to develop preliminary data that we can then apply for grants with the larger institutions, NIH and whatnot, and then create data that we can all review and learn from. And so this is a true testament to what patient advocacy and seed funding and what you all are doing is. Darla and others have talked about the mechanical control of voice, the 1, 2, 3, right? Respiration, phonation, and resonance. And this is a complicated process. This requires a lot of control of a lot of body parts. The lungs, the chest, the diaphragm, the intercostal muscles that control that allow us to take in a big deep breath, to push air through the vocal cords that we've seen a few times, and then to kind of process through this region here, the tongue. If it's malfunctioning or moving differently, or if you have a sinus infection, resonance is going to change. And so there's a lot that can go wrong mechanically that can affect voice. And certainly your clinicians need to evaluate that and think about that when somebody presents with a voice disorder. But more importantly, again, in my own bias, this is all controlled at the higher level, the brain. The brain sends signals to all of these muscles, the respiration muscles, the laryngeal muscles, the voice, the vocal cords or vocal folds, muscles that are involved in the resonance. And this is an oversimplification for sure, but there is a lot going on in the brain. There's a lot of electrical signals flying from one region of the brain to another. I'm going to spare the details of this, but page 19 of Our Voice, the newsletter that I think everyone received today, has a good description of kind of the brain regions that are involved in voice production. You don't have to read that now, but for your homework tonight, I think it'd be a good idea to learn more about that. But those are the components of what I'm interested in studying. And so as a neurologist, as someone who has tools, neuroimaging tools, taking pictures of the brain, we're able to study these networks and how this communication occurs in normal voice, and then of course in voice disorders that result from brain dysfunction. So we've learned a lot about spasmodic dysphonia. I'm going to focus a lot on this, and we've heard today there's some interchange between the term spasmodic dysphonia and laryngeal dystonia. The bottom line is that there is a strong level of evidence that this is a neurologic disorder. Dystonia is defined here: involuntary, sustained, or intermittent muscle contractions causing abnormal movements, postures, or both. So in the AD ductor versus AB ductor, what's happening is we have an involuntary, sustained contraction of these muscles here that either close or open those vocal folds. And that's, I'm going to argue, is controlled by the brain. And so that's important to understand. This is, it's fitting in line with the concept or the construct that this is dystonia. It's task specific. You've heard this again. It only occurs when you speak, and it only occurs when you speak in certain scenarios. It's less affected when you're whispering or possibly singing in spasmodic dysphonia. It's not occurring all the time because otherwise, if those vocal cords are closed, you can't breathe and you die. We've heard that a few times. So fortunately, that's the case. And the reason I mention that is many other forms of dystonia, hand dystonia. So dystonia can affect one body part. It can affect multiple body parts, the voice and the neck, the voice and the hand. It can affect the hand alone. Writer's cramp, I don't know if anyone has any friends or family or anyone that has writer's cramp. That's a form of dystonia. It's task specific. Only when people start writing do they start having cramping or curling of the hand that prevents them from writing appropriately. And so that's a very unique feature to dystonia that gives us a clue that this is a neurologic disorder. And then I already mentioned it's laryngeal dystonia or spasmodic dysphonia is associated with other forms of dystonia. We've heard it where people have neck involvement and the voice involvement. So that's a clue that these are related and result from that. And then I'm going to focus a lot on neuroimaging, right? Taking pictures of the brain, understanding what's occurring in the brain of people with various forms of dystonia, including spasmodic dysphonia. And I guess it's worth, along those lines, we've heard about Christina Simonian here. This is a picture that was obtained using PET technology. We inject a dye of sorts into the blood. That dye binds to a specific chemical or a receptor in the brain. In this case, it was a dopamine receptor. And sure enough, dopamine is affected. The processing, the information, the neurotransmitter, the chemical messenger in the brain, dopamine, is affected in patients with dystonia. And so that's additional evidence that is going to kind of guide us in how we're thinking about this. Unfortunately, replacing dopamine or taking dopamine away in people with laryngeal dystonia doesn't fix it. So while we understand this, we know this, we still need to kind of plow away at this and understand this better. So this is an important concept. I've thrown this in here. This general concept that brain location typically equals brain function. If someone has a stroke, injury to a region of the brain, the Broca's area, for instance, they have difficulty producing language. If someone has a stroke in a region of the brain that controls the hand, they can no longer control the hand. It's weak. This is reciprocated in the context of Wilder Penfield, who, thinking about advancing technology, he's a neurosurgeon. He had folks in the operating room for various surgeries. These folks were awake, and he stuck an electrode into regions of the brain, and he marched right down the motor strip. So we know that this region of the brain here controls motor output, and he stimulated. He sent electricity in these regions of the brain. And sure enough, when he was up here, the foot wiggled. When he moved down here, the hand wiggled. When he moved down here, the face wiggled, right? So this correlates the action, a structure-function relationship. We know that this region in general is going to cause this outcome. What we didn't see in these experiences is when he didn't have a particular region of the brain that he stimulated, at least that we know of, that moved the vocal cords, right? And that's a harder experiment. But the concept being that there are typically brain locations that associate with functions, and those are very discrete. Localizing brain function is important, right? So we have tools, electric stimulation on the opposite side, using imaging, neuroimaging, MRI, CT, to start to shine spotlights into regions of the brain that are really important, that control different aspects of function. And when we shine that spotlight into these various brain regions, and we understand what is controlling speech, for instance, we first have a better understanding of what's going on, right? How is the brain organized? When we know how something's working, we can start to tinker with it and understand when it's not working, what's going wrong. This really allows us to target research. As we go through each iteration of research and we learn more about any disease, spasmodic dysphonia, we can start zooming in our microscope, so to speak, and start looking. We start at a gross level. We look at the brain structure. We understand that a specific region is affected. We can start dissecting that area, so to speak, and really putting that under a microscope, understanding what are the genetic factors, what are the chemical factors, the neurotransmitters that might be malfunctioning in that region, and how can we leverage that to better treatment. We can improve clinical applications, right? We've heard that a clinician, eyes and ears are really the only way, the best way to diagnose spasmodic dysphonia right now. Can we come up with a tool? Can we put someone in a CT scan, an MRI scan? Can we take blood from them and make a better diagnosis? Understanding this brain function and dysfunction is going to allow us to do that better. And then, of course, we can create better therapies. And that may be through medication, right? That's the goal. I think everyone would agree that if we can take a pill every day, that might beat a needle to the neck. That's a goal. The deep brain stimulation surgery is, as we've heard, a very promising therapy. We don't fully understand if it worked well in laryngeal dystonia. I'm going to talk a little bit about that, or spasmodic dysphonia. TMS is transmagnetic stimulation. So this is a relatively less invasive procedure where there's a device placed on the head and it can send electrical current or pulses into the brain and manipulate the brain, creating either an improvement or dysfunction in some cases, and can be either a tool in research but also can be a therapeutic tool. And so our goal is to improve all of these with our research. I mentioned this already. So the concept that structure equals function in neurology is a strong one. Generally speaking, one brain region equals one function. Dystonia is different. In the 70s, David Marsden and others they looked at people who, again, had a stroke, right? There's injury to a specific region of the brain, in this case highlighted in red here. It resulted in dystonia of some type. More commonly than not, when folks have a stroke, injury to the brain, and develop dystonia, it's in this region of the brain, the basal ganglia, but not always. In fact, many areas of the brain, the basal ganglia here, the thalamus, a relay station deep inside the brain, the cerebellum, and again, page 19, 20, and maybe 21, it might be a long article in our voice, will describe this and how this is important. But the concept being that it's not just one region of the brain that's involved. It seems to be the interaction between numerous areas of the brain. And this is an important concept that has now allowed us, with tools that we've developed, MRI, to study this. And so magnetic resonance imaging, MRI, is a tool. It's relatively accessible, for better or for worse, and I'll talk about in that second. It's non-invasive. It doesn't hurt. The biggest risk of being an MRI is if you're claustrophobic, that can be discerning, but boredom, right? Lying in the scanner can be boring. And that's not a bad risk. That beats needles and wires and electrodes and stimulation. But what we want to do with magnetic resonance imaging is further explore the structure, right? What are the brain regions that are involved, and the function? What are the regions of the brain? How do they communicate that contribute to voice, normal voice production, and how is that potentially abnormal in spasmodic dysphonia? I'm not going to go through the details of this for time. The general concept is we can use this MRI tool, right? So the patient lies here, the head goes in there, we take pretty pictures here, and we can measure the energy levels that are occurring in the brain. The idea being that regions of the brain that are active have a higher level of energy, and we can look at that either through levels of oxygen or glucose is really the primary measure there. And regions of the brain that are involved in a similar function, voice production, for instance, if we draw, we take pictures of the brain, we take a picture of the entire brain, and we draw a little circle on a region that we think is involved in voice production. The energy in a person who is resting in the scanner, not doing anything, is going to fluctuate very similarly to other regions of the brain that are involved in that same process. And so this technology is called resting state functional MRI. When patients are resting in the MRI, we can take pictures of the brain and understand which regions of the brain are functionally connected. And so we can start mapping the brain with respect to that. And to a point of this, laryngeal dystonia or spasmodic dysphonia is particularly intriguing to study in this state. And the reason is because there's a lot of interpretive caution that needs to be held within MRI technology. When patients move, as I've tried to depict here with this head watching off the water, I couldn't find any better pictures of head movement than that, but I think it depicts the issue here. When patients move, or participants move in the MRI, it can create confounds of interpretation, right? So the signal that occurs in the MRI may be because the head's moving and we're just measuring a different intensity level, blood level, a different region of the brain because it's moved in the bore of that magnet. It may represent the voluntary control of, boy, I've got some water on my head and I've got to move. And it could be that conscious component, et cetera. So we really want to avoid that. Many studies using this technology in MRI have not accounted for this problem. And so a lot of research has demonstrated potentially, very highly likely, false positive results. And I'm not going to, again, in the lieu of time, I'm not going to go through the results of this, but this is something that our group demonstrated, that when we account for that movement, either keeping the patient still or statistically eliminating that, then we get much more close to the truth as far as what's really occurring in the brain. And this is a really important feature in when we start to hear media reports, we found something in the brain and the function, is that as a critical scientist, I'm going to go back to that study and make sure that they've accounted for this head movement. That's a really critical aspect and a problem in this field of science where MRI machines are available everywhere and people are doing this type of research everywhere. And so our research group has asked a question. We created a hypothesis. In research, we create a hypothesis, right? We think about the literature, what it exists, and we make a point. We say specific areas of the brain have abnormal communication, abnormal networks in patients with dystonia, including spasmodic dysphonia, that affects different body regions, right? So the idea being that there is going to be a potentially single region in the brain that dysfunctions in patients with spasmodic dysphonia, cervical dystonia, limb dystonia, and dysphonia that affects the face, the eyes, cranial dystonia. And so to study this, we in our lab studied high quality functional MRI data, again, accounting for the movement across a large population, the largest population that's been studied. 56 patients were included in awake resting participants with various dystonia subtypes, spasmodic dystonia, cervical dystonia, limb dystonia. And we compared that to normal controls. And in addition to that, we used what we knew. We knew that this deep region of the brain, the basal ganglia, we've just zoomed in on the basal ganglia here, has dopamine dysfunction that seems to contribute to dystonia, spasmodic dysphonia. We drew a circle around that in these patients that we scanned with the fMRI, and we looked at where, in relation to this, there were differences in the communication between that particular region of the brain in those who had dystonia compared to healthy controls. And sure enough, we were able to find an area, multiple areas, deep in the brain, in the basal ganglia, that had weaker brain connections in patients with dystonia, including spasmodic dysphonia. So this really supports this notion that the basal ganglia is important, that dopamine may play a role in creating this inability for the brain to communicate with other regions in that component. And so this is important. This allows us to shine yet again, the spotlight in this region. We weren't wrong. We're not wrong. This isn't wrong. Dopamine may play a role. It shines the spotlight in that. We've validated that. But since replacing dopamine doesn't fix it, now we've got to investigate further. This concept that dopamine is important is not the complete story. The way that dopamine is processed in the basal ganglia, it acts as the brake and the gas pedal with respect to movement. And depending on whether the dopamine binds to one receptor or another, D1 or D2 to simplify it, which is what it is called, D1 receptors versus D2 receptors, press on the gas and move, or press on the brake and stop. And so the thought being that there's dysfunction, there's imbalance in dopamine and how it binds to those D1 or D2 receptors. And for instance, in spasmodic dysphonia, the gas pedal's pressed too hard, right? The brake pedal doesn't get pressed enough. And that imbalance contributes to that excessive movement of the vocal cords. That dopamine in the brain is affected by other chemical neurotransmitters. Acetylcholine, for instance, we can measure that again, going back to that technology of PET. These are normal, healthy controls, but we can see that acetylcholine levels are extraordinarily high in this same region of the brain we're interested in, the basal ganglia. And so this is a big clue with respect to this. And this is something we're pursuing in our lab. Sure enough, this region that was discovered in 1970s, early 80s, when we take a picture and compare healthy controls and those with dystonia, including spasmodic dysphonia, we see a difference of acetylcholine levels in this region of the brain. This is new. This is hot off the press. This is important because there are now new medications that could potentially target this. This is still early. This still needs to be studied. We do know that anticholinergics, medications that decrease acetylcholine in the brain, do help people with dystonia, some in spasmodic dysphonia as well. The problem with these meds are they're not used much at all. I doubt anybody or many in here are using these medications because they create extraordinary side effects. These medications don't just bind to this region, but they bind to all these other red areas in the brain, and they depress that, and they cause people to be confused. They cause people to have difficulty peeing, have difficulty pooping, being sleepy. Those are major side effects that aren't tolerated. But that's because we haven't honed this medication, these anticholinergics, to really target the specific region of the brain, the basal ganglia, where we want them to. These groups at other places have really honed in on this and allowed us to create drugs that may be more specific to that region of the brain and have an influence. So this is a line of research that shows a lot of promise, perhaps the pill that we need. Another kind of direction, so taking away from the biochemical and the molecular level, is this concept of the group average studies, right? So what I've shown, that hypothesis that there's a specific brain region that's involved in dystonia, involved recruiting lots and lots of people with dystonia, studying them all, lumping them all together. Guess what? All of you are different in here. All of your voices sound different. And so maybe looking at group averages and averaging data across it isn't the best idea. Group averages give us a really pretty map, right? On the left here, this is a group average of functional connectivity, right? So yellow represents a particular network that controls one thing, purple controls another function in the body. When we blur, right, all of these images together, we stack them on top of each other, we average them, we get a really pretty map. And so point number 1 and point number 2 in this case are in this yellow network. These 2 points in the group control the same function. But when we look at one of the individuals that we use to look at this map, point 1 and point 2 are in the same place in the brain, but point 1 now is in a purple network. That controls a different function than it did here. And so this person, if we're sticking an electrode in there, for instance, to treat that person or targeting a drug to that region, it's not going to work for that individual. That's a problem, A, because if we're including this person in our clinical trials, right, to understand the drug, and it doesn't work, it's going to take away from the others that it's going to work in. B, right, we're just targeting the wrong place, and that's not going to be fun for any individual. So this is something that Dysphonia International has really helped us with in funding this and thinking about this in the individual level. How do we use this technology of MRI to start to advance the individual approach to this? And so I work very closely with Evan Gordon. He's a doctorate student who's really worked on this method here, precision functional mapping. Again, same technology, MRI. The risk here is that you're even more bored because you have to lie in the scanner longer, but to get more quality data. And by getting more quality data, we can go back to what Dr. Penfield did in the OR, and we can look at these regions, right? Foot, hand, tongue, in that motor cortex, foot, hand, tongue, in 10 different individuals, 1, 2, 3, 4, 5, etc. While the general pattern is the same, where foot, hand, and mouth are represented, there are individual differences in the extent of the regions in which that's involved and the distribution of that. And of course, when we average it all together, it creates this nice, beautiful, robust signal where foot, hand, and mouth are very big. If we're targeting it with electrode, we would think that we stuck it in anywhere in this area, we would get the effect. But the reality is probably not in this one where this region for hands is much smaller. And so this technology allows us to really zoom in on the individual level. Very cool stuff. Hot off the presses, relatively speaking, is this Penfield experiment, electrical stimulation. Let's march right down, and seed in these MRIs, what we thought was foot, hand, mouth components, and effector, meaning that when Penfield stimulated this region, number 1, number 3, and number 5, he got a foot to twitch, he got a hand to twitch, he got a face to twitch. But in between these regions, which Penfield never demonstrated, is another network. We never knew this before. This new network is interesting because it spans between the foot, the hand, and the mouth component. And when we seed this region here that's between foot and hand, it also lights up the region that's between hand and mouth and down here. And so this seems potentially to be a network that might coordinate the movement across body parts and be involved and important in this. And so this is a really important concept. Dr. Gordon and his colleagues and ourselves call this the SCAN network. And this is because it affects the higher network of function. Even hotter off the press, not published data yet, I grabbed this when I was running to the plane yesterday, is this concept that this occurs in dystonia. So when in kids, pediatric patients who develop dystonia as a result of whatever disease it is that they get dystonia, when they use this application, precision functional mapping, in fact, it is the SCAN network that's involved, not the effector regions, not the typical Penfield regions that we thought of that might control movement in the motor cortex. This has huge implications because now we've shined a spotlight on our new regions, right? And when we apply TMS, transmagnetic stimulation, we put an electrical current on the skull with the intent of stimulating that motor cortex. Maybe we were just aiming slightly at the wrong place. And if we move another centimeter or two, we have potential of treating this. So this is a big deal. I have, in addition, applied this specifically to patients with spasmodic dysphonia, this concept of precision functional mapping. And so the general concept being we put someone in the MRI, they're bored out of their mind, but they're happy to be there to be contributing, and we're grateful for that. They speak in the scanner. When they speak in the scanner, we measure that blood flow or that energy consumption, and we see regions of the brain that are involved in the voice production. When the vocal folds are activated, it's a result of these areas, in the brain being activated. And so we can then use these regions that are involved, that are important for voice production, for allowing those vocal cords to move properly, as seeds to then collect. Well, let me back up a little bit. Before we apply them as seeds, we can replicate that in multiple participants. So, participant 1, right, voice network here, participant 2, voice network here, participant 3, voice network. A little bit different. General pattern is the same, but a little bit different. And so again, if we're averaging this across the way, and this region here where this is real robust here but not present in here, may not come out in those group level averages and studies. And we may, again, be stimulating the wrong place. This technology is also really cool because as opposed to Dr. Penfield, who stimulated electrically on this area, this is a picture that really takes a slice way deep in the brain. And so we can trace the path, the energy, the electrical activity through the brain, deep into the brain, into the basal ganglia. So this is that basal ganglia that keeps coming up, that same slice that Dr. Marsden demonstrated early. And even more importantly, right, participant 1, robust signal. Participant 2, maybe a little different place and less robust. Participant 3, a little bit different. And again, if I'm sticking an electrode in this area or this area of the thalamus, which is commonly done, I may miss that in one person versus another where that moves. Now let's take it a step further, right? We've heard about how Botox might trick the brain, right? It kind of rewires the brain. It creates differences in how the brain processes information. Let's do that, all right? Let's measure the resting state functional connectivity before we trick the brain. Let's see what it's doing. Let's inject Botox. Let's have, when the Botox is working, have the participant come back and scan it again. Does the Botox manipulate these brain networks? Is the brain different when the voice is working better? Presumably yes, right? I'm arguing that the brain is contributing to this disorder, and with successful therapy, the brain should change with respect to that. And in looking at that, we may have targets for better therapy, knowing that those particular brain regions are improved with success with Botox. And sure enough, right, so these are the same pictures I already showed you. When a person vocalizes the MRI, this area lights up in participant 1 here and 2. And this line shows the difference in that pre-Botox state compared to the post-Botox state. And so what this reflects is in these same regions that are activated by the voice, a difference in how much it's activated in a successfully treated state. And so again, this gives us a potential target to really aim at with respect to all the things we do, with respect to the research looking at the chemicals, with respect to the potential therapeutic implications of sticking a wire there and having success with that. And so this is why this is important, right? We can start to really zoom in, dissect that area, understand what are the chemicals, what are the medications we might be able to produce in that, how might we send electricity into this region, and those who are brave enough to do this, to have success, not to tell somebody, hey, it might be a 50-50, a flip of a coin, whether you have any improvement at all. And of those who improve, it's sometimes very variable. And then again, TMS, it's probably more appealing to have a little stimulator on your skull rather than have a hole drilled in your head. We're trying all of these things. We want to make sure that we have all the options available. And this neuroimaging allows us to understand how the brain is malfunctioning, dysfunctioning in spasmodic dysphonia, and shining that spotlight to allow us to expand research. I understand it can't happen fast enough, but it does take this knowledge to really advance the technology, to really advance this. And we are working very hard to do that. So I want folks to know that. In summary, I'm almost done. Lunch is almost here. Patient advocacy is critical. Thank you for being here. Spasmodic dysphonia reflects brain dysfunction. Specifically neurotransmitter imbalance, historically dopamine, now acetylcholine, possibly other receptors, GABA being one of those with respect to alcohol and the questions that alcohol have. That's relevant and influences dopamine as well. Neuroimaging, right, the methods that we employ using these advanced MRI methods demonstrates that there are common network or circuit level dysfunction across various dystonia types. That gives us a really good spotlight at a group level. There is a new network that coordinates movement and is affected in dystonia, and we really need to start zooming in on that and studying that, and that shows a lot of promise. And we are now able to study individuals, patients who are affected by this, and really to hone our treatment efforts with respect to that. And so this has huge treatment potential. I'm optimistic. I continue to wake up every day excited about this and come to work to understand this and to learn more about this. This is where I hail, St. Louis, Missouri. That's Forest Park down here. This is our group, a movement disorder neurologist, a wonderful group who are all fighting for this and trying to help. So that's all I have to say. I think with 3 minutes to spare before lunch, perhaps, I'm happy to answer questions now, lunch, and beyond.